All right, thank you everyone for joining our latest iteration of the Singapore Single Cell Network meeting series. Sorry for the slight delay. We're using WebEx today. Some of you might be on Teams, might have been on Teams, but it's WebEx. Two exciting technology speakers with us today. We've got, I'll introduce our first speaker for starters, Lih Feng Cheow, and Jiang from Vizgen will be our next speaker. So Lih Feng is an assistant professor at NUS Biomedical Engineering and is a PI at the Institute of Health and Innovation Technology, iHealth Tech. Used to be my colleague and collaborator at the Genome Institute of Singapore, renowned single cell technologist. Got his bachelor's actually in electrical and computer engineering from Cornell and PhD again in electrical and computer science from MIT. And his interest in developing tech platforms to perform precision medicines of multimodal precision measurement of single cells, like say genetic, epigenetic, transcriptomic, and innovative technologies for biosample preparation and using all those for disease diagnostics. Lih Feng, great to have you here.
Okay, thank you, Shyam, for the kind introduction, and thank you, Vizgen, for organizing this seminar. Right. Today I'm going to talk about some work that we have completed in our lab recently. And the title of my topic is Integrative Single-Cell Transcriptomics and Functional Profiling with TRAPS-seq and PAINTKiller-seq. I'm assistant professor at the Department of Biomedical Engineering at NUS, National University of Singapore. And I'm also a PI at the Institute of Health Innovation and Technology. Okay, just give a very brief introduction of the things that I do in my lab. Right, we are called the Technology Innovations and Systems Biology Lab, and there are three research focus in our lab. The first one is, of course, single-cell technologies, and we're going to talk about some of the topics in single-cell technologies. Interesting works that we have worked on recently. The second team in my lab is actually one single molecule, right? And in this team, we are actually developing very novel digital PCR-based platforms to perform all sorts of multiplex measurement of our molecules. And lastly, we also very interested in epigenetics, particularly in DNA methylation. Over the years, we have actually developed a few methods to look at DNA methylation both in single cell as well as in cell-free DNA using informatics. But today I'm going to focus on single cell work, in particular, two of the recently published work on the TRAPS-seq and PAINTKiller-seq. So the first thing that I'd like to just kind of introduce is that the cells in our body are very complex and they do not exist in isolation. In fact, they interact with each other very persistent. And one of the best examples is in the immune system, right? Our immune system consists of different immune cells, the T cells, the B cells, and K cells, right? And they actually work together, right? They are different cell types. They actually work together to really kind of make this very complex system. And when I mean work together, they are actually constantly talking to each other. They are secreting cytokines, they are secreting chemokines to each other as well as the other cells. Right? There are also other interesting functions of immune cells, such as certain classes of immune cells like cytotoxic T cells and NK cells, which are capable of recognizing a target cell and actually killing them, right? That's really very fascinating topic and really a lot to be learned, right? By being able to look at the functions of these systems. So in the past 10 years, past 10, 20 years, there is really an explosion of technologies to be able to profile these cells at the single cell level. First of all, we have the single cell surface protein measurements, and you have the well-known flow cytometry, right? where you can actually use fluorescence to measure these molecules based on antibody binding, and you can very effectively measure the surface proteins in a very multiplex manner. On the other hand, you also have technologies such as TRAPS-seq, 10x Genomics, utilizing droplets microfluidics that you're able to now perform reaction for single-cell transcriptomic measurements, right? And this actually allows you to look at whole transcriptome. People are able to look at chromatin accessibility, various epigenetic factors as well. Right? And you also have combinations of both techniques, right? If you're able to kind of put antibodies with barcodes, you're able to actually combine surface protein measurement as well as single-cell transcriptomic measurements in technologies such as SSCN, which allows you to perform multimodal measurements. But one thing that is really missing from this picture is about the function. So we have all these measurements about the basic molecular parameters of the cells, be it protein on the cell surface or the transcriptomics. But what are the functions of the cell, right? Are they able to secrete proteins of the interest when need to? Are they able to kill cells? Right? So I think this is a big missing picture in this whole puzzle here. The cell functions are really very important. Right? Take for example, recently people are very interested in looking at immunotherapy, right? And people have found that the polyfunctional T cells that you manufacture during cell-based immunotherapy are a very important factor to determine whether the patient can actually respond favorably to this therapy. And people have actually performed these measurements. Right? They have made use of microfluidic devices and arrays of antibody strips to be able to measure protein secretion. Separately, they're able to perform single-cell RNA sequencing of these cells, right? And they're able to get surface protein secretion. They're able to get the transcriptome of single cells, but there is still a lack of correlation of what are the molecular parameters of a single cell that actually cause the functions of these polyfunctional cells. Right. So this is the gap that we're trying to address. And then in the next few slides, I'm going to describe the technology that we developed in our lab, which is known as TRAPS-seq, which stands for Time Resolved Assessment of Single-Cell Protein Secretion by Sequencing. The basic idea here is that we are able to link the single-cell secretion with its transcriptome, and we will be able to see what is actually the transcriptomic signature that correlate with particular functional outcome itself. So let's look at what are the challenges to be able to do this. So unlike surface protein, the proteins that are secreted by a cell, you actually quickly diffuse away, right? When you just let it be in nature. So in order to associate a secreted cell with the particular cell that was secreting it, right? We would need a method to be able to link them together. So the approach that we take is that we attach some capture antibody, and in this case, mostly antibodies, on the cell surface, so that when this cell is secreting a protein, right, this protein does not diffuse away. Instead, it actually gets captured on the cell surface by this capture antibody. The secreted proteins that are captured on the cell surface can then be profiled in the same way as SSCN. The implementation of TRAPS-seq is really straightforward, right? The only thing is you need to do a very generous cell modification that maybe takes 30 minutes to an hour, right? What you do is you actually take these cells of interest, right, the protein secreting cell, and you actually functionalize them with capture antibody. In this case, we make use of a bispecific antibody. One end can attach to the cell through some ubiquitous surface markers such as CD45. On the other end, you have the capture antibody, and we can actually have multiple capture antibody against multiple secreted proteins. And after that, you could stimulate the cells, allow them to secrete the proteins of interest, and these proteins would be captured by the surface antibody, and finally you come in with an orthogonal detection antibody with a barcode to be able to stain and label these protein secretion. Finally, you perform single-cell sequencing using platforms such as 10x Genomics, and you can simultaneously measure the transcriptal, the secretal, as well as at the same time using this method. So the first thing that we need to verify is that the proteins that are captured by the cell are indeed the cells, the proteins that are secreted by themselves, and not proteins that are secreted by other cells. So in order to do that, we perform very stringent control experiment populations of cells, for example, Jurkat cells that do not secrete anything, T cells that do not secrete anything, but they start to secrete significant amounts of IFN gamma, IL-2, and TNF alpha upon stimulation. Right? And here the facts plot show exactly that. What we do next is that we perform a mixture of these Jurkat cells and stimulated T cells in the same pot. And we see that the Jurkat cells do not capture any of these cytokines, but similar percentage of cytokines are captured in the T cells. Right? So this experiment really established the specificity of capturing self-secreting proteins in this single cell. So with this, we apply these TRAPS-seq or PBMCs, right? We focus primarily on T cells because these are cells that secrete these three cytokines. Right? And because we are able to measure both the secreted protein as well as look at the transcriptome, we can perform correlation between mRNA and secreted protein. And here what we saw is that there is different kinetics and limited correlation between the mRNA and secreted protein, meaning that the mRNA cannot always serve as a perfect proxy of the secreted protein. We also see different kinetics, particularly the RNA actually expressed first, followed by the proteins, and it is related to the translation, the finite time scale that is needed to translate the RNA to specific proteins. So the second thing that we look at is to see whether whether these different protein secretion patterns actually correlate to specific cell types. And because we can look at the surface proteins, right, we can correlate them with the secretion pattern. And here we see that the different cell types as marked by the surface protein do actually correlate with different secretion patterns of these cells as mentioned by TRAPS-seq. Right? And you can see that the CD4, CD8, T cells, and the memory T cells, they all actually have very particular secretion pattern. And finally, using this correlation, we can perform some discovery work. Here we focus on the triple positive cytokine secreting cells, which are known to be quite important in immunotherapy. And what we found was that if we actually select for cells that express high levels of CD95 and CD69, we get a significant enrichment of triple positive cytokine secreting cells. So we can also correlate this cytokine secretion pattern with the transcriptomic signature. And what we saw here was that for different kinds of cytokine secretion pattern, they really correlate with specific transcriptional modules. We can actually perform kind of gene set enrichment of this transcriptional module, and we saw that, well, the different cytokine producing cells, right, actually make use of different mechanisms. Right? They are enriching certain mechanisms that actually correlate with their particular signature. So this is actually very useful for people to kind of know how you can actually engineer the cell, right, or actually promote particular secretion patterns of the cell for functional studies. And lastly, we found that we can actually do something that is very, very interesting with TRAPS-seq. Traditionally, when you're looking at site-seq or when you're looking at single-cell mRNA sequencing, you're actually looking at the snapshot of the RNA or the proteins because you lyse the cell and you're measuring the RNA or the proteins at the same time. But the function of the cell really is not static. They are really dynamic. The cell could be secreting certain proteins at one time and stop, right? Fast secretor in slow secretor, right? So with TRAPS-seq, we're wondering whether we can measure the protein dynamic, and we found a very easy way to do that. And this is shown in the diagram below. First, we functionalize the cell, just as we have done with the static TRAPS-seq. We allow the proteins to be secreted, and we perform a first round antibody labeling. Subsequently, we allow them to secrete in the second time period. And now we actually capture additional proteins, secreted proteins, and this would be identified with a second round of antibody labeling with a separate barcode. So by looking at the barcodes of the antibody, you can identify whether these cells were secreted in the first time period or the second time period. So now we have this additional information, cell secretion dynamics, not just cell secretion static measurement. And we can do a lot of very interesting measurements of that. First of all, we can measure the cytokine secretion dynamic. We can see what are the transitions from particular cytokine secretion states, for example, triple positive cytokine secretion state to specific double positive cytokine secretion state or single positive cytokine secretion state. We found that these kind of dynamics are not random, right? In fact, they are very much preferred patterns of cytokine transition state. And what we found was that just by looking at this, right, we can come up with a hypothesis, which is matching with the measurement that actually TNF alpha is the cytokine that's extinguished the most quickly, right, followed by IF and gamma. Lastly, IL-2 is the most persistent cytokine in every case. And using this, we can actually predict, right, what are the changes in cytokine secretion dynamics in cells. We can also use this protein secretion dynamics, right, to correlate with the particular cell type or with surface marker or by looking at the whole transcriptome, we can actually look at certain pathways that actually correlates with particular protein secretion. Okay. So that's the story of TRAPS-seq. Because of time limitation, I'll be able to present more, but I'd just like to introduce to you this technique that we have, the methodology that we have developed, right, and some of the basic analysis that we can using this particular method. In the remaining time, I would like to move on to the next technology that we have developed in the lab that I would like to talk about, and it is on painkillers. So the other cell function that is very important in cells is actually cell cytotoxicity. Certain immune cells, such as T cells and NK cells, are able to recognize target cells and kill them. But typically what you do is that you mix a population of immune cells, mix a population target cell together, and you see how many of these target cells are killed without knowing which of these immune cells are responsible for killing them. Are they all equally capable of killing these target cells, or are there some specialized immune cells that are capable of killing multiple target cells? So that's a very interesting question that has a lot of clinical applications as well. So in order to solve this problem, we were inspired by kind of a forensic approach. If you look at this cartoon here, if you are police trying to identify a murderer, one suspects, right? But one clue that you could get is that this particular person has blood stains on his body, right, then he is a very likely suspect, right, because probably the blood stain from the victim which is splashed on him, right, during that killing activity when they're in close proximity. So this is the kind of concept that we take, that we introduced and implemented in PAINTKiller-seq, which is the full name is proximity affinity intracellular transfer identification of killer cells. So in order to implement PAINTKiller-seq, we need actually two main criteria. First of all, the target cells need to have a sort of blood, right, that can be released and can actually stay the killer cell. And secondly, the killer cell need to have kind of a sticky coating, right, to capture any of these blood stains from the target cell. So to achieve the second aim, what we did was very similar to what we did in TRAPS-seq, which is that we attach antibodies against these intracellular proteins, which is anti-CFS, on the surface of the killer cells. And secondly, to introduce the blood in the target cells, we incubate them with dye or CFSE, which can passively go into live cell, and they actually stay within them. So they are intracellular fluorescent. So what happens is that when effector cell actually lies on target cell, right, these CFSE that are now found in intracellularly, right, will be released, they will leak out, and they will actually be captured by the killer cell. Right? The killer cell that does not lyse any target cell, right, will not capture any of these CFSE. And finally, to make it compatible with sequencing, we can label them with antibody. So we also do this specificity study, right? First of all, we look at the basic principles of this particular painkiller assay. We took primary NK cells, which are going to kill K562 cells, right? We incubated them together, and the fact plot on your right shows that if when you with time, you see more and more of the cells, of the killer cells, of the primary NK cells actually gaining this CFSE signal, meaning that they have killed. But interestingly, not all of the killer cells are killing. We also look at the specificity. We mix the primary NK cells with K562 cells, and we also put with them some bystander cells, which are primary T cells, which we also put on the coating. And what we saw was that the NK cells actually gain this CFSE signal, but the bystander primary T cells actually remain very clean and do not gain any CFSE, meaning that this staining is killing specific. Right. We could combine the cell killing, cytokine secretion, which is TRAPS-seq and surface protein measurement together, right? And here what we are showing is that we are measuring the cytotoxicity, the secretion of IFN gamma, right, by also attaching anti-IFN gamma of the cells. And we are also measuring CD107, which is a marker of killing that some people use. And we are able to see all these three parameters together. We are able to identify cells that can simultaneously secrete cytokine and kill, right? And we were able to see the dynamics of killing and cytokine secretion. We saw that the killing happens much faster than cytokine. And interestingly, we also see that this killing is in kind of a monotonic increasing fashion, whereas the CD107A increase and decrease, and we think that this may be due to the instability of the CD107 protein. Finally, we applied this technology, PAINTKiller-seq, to look at primary NK cells, what are transcriptomics, as well as cell surface determinants of killing. We are able to get this view map here, which shows the different subpopulations of primary NK cells, and you see the cluster 2 actually are highly enriched for cells that are killing. And when you look at the gene expression of these killer and K cells, you see that they are also simultaneously enriched for genes such as IFN gamma, which we also saw previously, so killing, and cytokine secretion. We see secretion of various chemokines as well. We could look at the whole transcriptome, look at marker genes that actually characterize this subpopulation, and find what are the gene expression modules that are actually activated. So with that, I like to end my presentation today. I hope I have managed to show you the technologies, the new capabilities that we developed in our lab. It's a very simple assays that can be used initially in different applications. And very happy if you have any collaborations or any ideas that we can work together. So with that, I'd like to acknowledge the people in my lab have done this work as well as funding. Thank you for your attention.
Fantastic. Thanks very much, Lih Feng. Managed to show two exciting technologies in a short talk. Everyone, please use the QA button in your WebEx to ask the question. There's a Slido button also, which I'm not familiar with, though I used it long ago. But yeah, I think the WebEx Q&A button should work. In the meantime, few questions from my side. I maybe start with biological question from TRAPS-seq. Actually, first, I guess I should say that most single-cell technologies, they focus on what's inside the cell or what's on the surface of the cell. So it's very exciting that you have gone beyond that to look at secretion. Mostly, at least my sense was always that you need to profile proteins in a tissue to understand secretion, but this is a very nice alternative that you can do in vitro. For the TRAPS-seq part, you mentioned that there was limited correlation between the RNA and the secreted protein.
Yeah.
And this is kind of in the tradition of more or less, for, I'd say 20 years, people have been talking about how the RNA and protein in a cell or a sample have limited correlation. And there's still a debate about, is that real, or is it just that the measurements are noisy, or that the time course is different or something? Because you yourself showed that there's time temporal dynamics in the protein secretion. And then, of course, there's measurement noise in any assay, and then particularly in single cell assays. Right? For example, if you look at RNA gene correlation in single cell assays, you'll see a very poor correlation between genes, even though you know that they are highly mechanistically related and co-expressed and so on, simply because the single cell assay has low sensitivity. Could that be explain some of the low correlation you found between RNA and protein?
Yeah, so I think that's a very interesting, that's a very important point. I think yes, measurement noise definitely play an important role, right? But I think for protein secretion, there is additional things that's coming, because we are actually measuring secreted protein, which it's arguably more functional, right? The secreted proteins need to undergo various different post-processing to get out of the cell. Some of them are made, they are translated, but they are stored in the cells, right? Some of them need certain post-translational modification to be able to target them for secretion. So we think that there are additional layer of regulation post-translation in terms of the secretion pathways that are actually important in this context of secretion. But of course, noise, translation dynamics are actually a factor as well.
No question. Yeah, there's definitely biological layers beyond transcription. Transcription, then is translation, there's first splicing, translation, post-translational modifications, export from inside the cell to outside the cell, and so on. And they will certainly reduce the correlation. I guess the question is, if you account for all that, right, how much is the real correlation?
Yeah.
Because and I'm asking that because it's a very important question. If you say there's no correlation, then why is anyone doing single-cell transcriptomics?
No. There is not a very high correlation. It is a positive correlation. I must admit that there is a positive correlation. But what the message I'm trying to bring across is that they are not perfectly correlation. So it still makes sense. Yeah, it still makes sense to be able to measure them because just measuring the transcription is not enough.
Absolutely. Good point. There's a question in the Maxine Lam. Thank you for using the Q&A button. Have you tried to isolate T cells from tissues with your and analyze them using your technology? Or are they too rare for analysis in your workflow? So will the technology, let's say TRAPS-seq or maybe even PAINTKiller-seq, perform when there's many different cell types present and so on? Okay, lots of questions. Let's start with the first one, if you remember.
Yeah, we have not tried this with T cells in tissues, right? But based on the principles of this, I presume that they will be applicable to tissue T cells as well.
Well, I guess implicit in the question is how many T cells do you need for your assay?
How many T cells do you need? So this, because I think ultimately we are actually making use of high throughput single cell transcriptomics platform. So that would be kind of whatever cells, the number of cells that you need for these platforms. That would be the
so like 10,000 should be good enough as long as it's at the right concentration.
That's right.
Okay, great. And will the assay still work if there are many cell types present?
Would the assay still work with many cell types? Yeah, so we are able to identify the different cell types by the transcriptome as well as cell surface marker. So in fact, that is kind of the selling point of this technology, to be able to work with multiple cell types.
Excellent. Question from Jiang. Jiang, thank you for joining. For TRAPS-seq, how many antibodies are on the surface of one cell? And will those antibodies get saturated if you have a lot of secreted protein?
Right. So I think we got asked this question a lot, right? We did kind of a back of envelope calculation. I think 20 would be no problem. And we have tried 3, but our back of envelope calculation tells us that 20 is not a problem. But a lot of this also depends on the secretion level of this antibody. If you have some certain proteins that are secreted at low levels, then you can presume targets into the same cell.
Okay, your voice is breaking up a bit. I don't know if it's my network or yours. Yeah, I got the gist of your response. There's a question via the chat. Oh wait, sorry, that's the same question. All right, and then I wanted to follow up also on how is TRAPS-seq available? How can people use it besides collaborating with you, of course?
Yeah. So I think the limiting everything is available except for hard-coded antibodies, right? Which we have actually provided the methodology to conjugate DNA barcodes towards antibody. And you could do that easily. But if you want to just try it out, we are happy to collaborate and send you some of our antibodies.
Okay, so for now we can do it mostly on their own with some reagents from you, and next year you'll license it to a company we've all heard of.
Yeah, hopefully if someone is interested in licensing.
Okay, a question about PAINTKiller-seq also. That's very clever too, and knowing which cell actually did the killing is really a very, very key question in biology. And until now, we've only had more indirect evidence, I would say, and less high throughput evidence. So this is fantastic to catch the killer red-handed. I wonder if the next step is to not only paint the killer, but also handcuff the killer, arrest the killer. So, so in fact, I can offer this suggestion to you for free, no IP claim. You can call it TRAPS-seq. But so you biotinylate the CFSE or whatever, so that then you can capture the killer cell with streptavidin, and then you don't have to waste money sequencing the non-killer cells.
Yeah. So I think that's really elegant solution. Elegant suggestion. What we have done, that we have not actually shown here, is that we have actually sorted the painkiller, the painted cells, right? Because we have CFSE and they actually are fluorescent molecules, so we can actually sort them with FACS, and we have done that and gotten some very interesting results, which is that we sorted these killer cells from our NK population, and we sorted the non-killer cells from our NK population, and we actually cultured them for 2 weeks, right? And we performed this assay again. We saw that these killer cells that we sorted out in the first place, right, still retain the superior killing activity. So that means that there is something involved, right? It's not just temporarily a transient increase of the killing ability, but rather a long-term kind of epigenetic the killer properties.
That's quite remarkable. Yeah, and I wanted to comment on the temporal dimension of your assays also. Again, something that gets overlooked in conventional single-cell profiling assays. They essentially just take a snapshot. So very exciting, and hope to be able to use all these technologies to understand cancer-immune interaction, autoimmune diseases, and other phenotypes. Thanks very much for joining us, Lih Feng.
Thank you.
All right. And now over to our second speaker. So as long-time listeners know, we alternate, we try to pair one academic speaker and one speaker typically from the biotech industry. And then so our biotech speaker today is Jiang, who's co-founder and senior director of scientific affairs at Vizgen. Very thrilled to have him here with us. So as I mentioned, he's a co-founding member of Vizgen and highly awarded, including Forbes 30 Under 30 in healthcare, Spotlight Scholar at AIF, Stat Wunderkind awardee, amazing. And he completed his postdoc at MIT. So we have two MIT trained people today with us, and a PhD at Harvard University in Xiaowei Jiang's lab. As you know, Xiaowei is one of the was the creator of the merfish assay that Vizgen is based on. All right, Jiang, over to you.
Thank you very much for the introduction, and very happy to be here. So I'm just going to share my screen now, and let me know if you're viewing the full screen now or not for the presentation.
Full screen, yes, we see.
Okay, great. Yeah, thank you. And morning, everyone. So so today I'm going to mainly talk about sort of the status and some of the applications in spatial biology. And as a life science company, Vizgen is actually pioneering in the field of spatial genomics. We commercialized the technology called merfish, and are actually one of the first companies to launch a commercial platform called MERScope to the market. So for today's presentation, I'm going to cover 3 parts. The first part is a very brief overview of spatial genomics and the merfish technology. And then I'm going to spend a few minutes walk through the workflow of MERScope. And then at the last part, I'm going to highlight some of the key applications of merfish technology as well as MERScope instrument, basically to highlight why spatial genomics is important and what kind of new insights researchers might be able to gain from this type of new tools. So we'll begin by a very brief introduction of the field. So as all of us are aware, biological systems are very complex, made of many different cell types intricately organized in 3D in different tissue types. So even though within the same organism, the genome more or less stays the same, but gene expression level may vary a lot. So over the past 2 or 3 decades, sequencing, such as bulk or single-cell sequencing, has transformed our way of understanding biology and allowing us to interrogate the gene expression at single-cell level. But it can only show us parts, because in order to do sequencing, we either have to dissociate the cells or destroy the cells to release the RNA and DNA, while on the other hand, cells inside the tissue are actually spatially organized, thereby we lose that very critical spatial context within cell. And it's only in the recent few years, the field of spatial genomics started to emerge and allowing us to now do true in situ multiplexed detection here. So that really started to revolutionize and actually transform our way of understanding biology. And as mentioned, as a company, Vizgen is one of the very first company to provide a true single-cell spatial genomics approach and platform called MERSCOPE to the market. And this is a platform that based on single molecule RNA fish imaging and allows simultaneous imaging of hundreds to thousands of genes in a single run with extremely high sensitivity and accuracy. The instrument can scan very large tissue areas up to 1 square centimeter sized tissue with down to 100 nanometer resolution, and there's no need for sequencing. So as a platform, MERSCOPE is built based on MERFISH technology. MERFISH stands for multiplexed error-robust fluorescence in situ hybridization. It was developed by our scientific co-founder, Shyam's lab at Harvard University and published in Science back in 2015. MERFISH was built based on single molecule fish. So this is an assay where fluorescence detect oligoprobes are used to label different genes of interest, and by counting the dots that fluorescence, then researchers will be able to know where those transcripts are and how many copies there are. But there is a big limitation for single molecule fish. Basically, to do multicolor imaging using single molecule fish is challenging because you have to use spectrally distinct dyes to tell different biomarkers or different RNA species. So oftentimes less than 4. So MERFISH breaks that limitation by combining with a binary barcoding system, an error-robust barcoding system, and that enables massively multiplexed barcoding. And I'm going to explain the principles a bit later. And then build on this, then it enables true in situ gene expression profiling. So as a platform, MERSCOPE is an end-to-end platform solution. It begins by selecting a list of genes for imaging and for profiling in situ. And as of now, we at Vizgen's commercial offering, we offer up to 500 genes, but they will be up to 1,000 genes coming very soon. Then with the probes and genes selected, then the probes will be assigned a specific set of barcode, and then the oligoprobes will be created. Then the samples will be hybridized with the probes, and then it incubates, and then it will bind to the genes of interest. And then after sample prep, the sample will be loaded into a flow chamber, and the corresponding reagent pack will be selected, and that will be loaded to the MERSCOPE instrument for imaging. So the instrument itself automatically takes the image and basically once set up and configures the experiments, you can walk away, and then it started to automatically transfer the data for decoding analysis, and the code can also be put to visualization software Vizgen provides, and thereby you can explore the data in situ, like what's shown on this slide. So this slide sort of very briefly explains the principle of how MERFISH works and what are the 3 key stages of MERFISH. So the first stage of doing MERFISH is to basically hybridize the sample with a set of unique encoding probes, and each of those encoding probes condense part of the barcode associated with each gene of interest. And by associating each gene with multiple probes, it allows us to actually have enough signal to be detected on the imaging system. Then the second step during sample prep is to do tissue clearing, we use a gel to remove any unnecessary components of a tissue that could interfere with the measurement, thereby reducing the background. So we have got a way to boost the signal, a method to reduce the background. Then the last step of the sample prep is to image the barcode and read out and decode them. So since each gene is assigned with a unique barcode, and in order to read it out, we use a sequential round of imaging as highlighted by the picture at the bottom here. So basically, we flow in different readout fluorescence detect readout probes in, and if there is a binding event to that barcode, then it will be show up as the fluorescence on signal at a particular site. Then we extinguish the signal and then flow in a second subset of readout probes. If there is binding, then there is fluorescent signal. If there's no binding, then it will be dark. So thereby, by repeating this readout the same procedure, it helps to build an optic barcode that basically fluoresces on and off. And using this optical barcode, then it can actually be used to match with a pre-existing barcode that we assign to each gene of interest. And if it matches, then we will be able to decode and figure out whether it's gene A, B, or C. And if it doesn't match, we could apply an error robust correction or whether to potentially correct it or reject it. And that essentially allows us to actually locate and quantify each transcript of interest in situ, as highlighted on the right. Each colorful dots now are different transcripts, and the shape here is sort of the cell membrane of the cell. And essentially, what that allows us to do now is in situ gene expression profiling from across the whole tissue to subcellular resolution. Showing here is the FFPE human ovarian cancer imaged by 500 gene panel. And a total of 174 million RNA transcripts were detected in this sample type on the left and highlighting 8 marker genes here. And hopefully, by just looking at 8 genes, you can already see this very beautiful and heterogeneous distribution of biomarkers across the tissue. Then on the right is the animation that highlights and shows the raw Murphy's data, where you can zoom in at different tissue regions and now turning on all 500 genes, and different color indicates different RNA species, and hopefully you can kind of see the distribution of RNA transcripts inside a cell. And now and only turning on 8 now and highlighting the distribution of unique genes. And Murphy's platform not only allows imaging of RNA by combining protein staining, it allows us to actually label the plasma membrane of the cells, and that allows downstream single cell cell segmentation and downstream single cell analysis. And with that, then researchers will be able to assign individual transcripts to each uniquely identified cells, and then with that perform single cell analysis. Basically, kind of is very similar workflow for downstream single cell analysis, like dissociated single cell analysis. So with that, you can kind of start to imagine actually with this type of in situ spatial transcriptomic imaging at single cell resolution, it allows us to do molecular atlasing to profile the gene expression in situ across large tissue. And then it also allows us to analyze intracellular organization of the transcriptome at subcellular resolution, characterize cell state and function to perform true cell atlasing. And at a larger scale, as cells are interacting with each other in situ, also now at the tissue level, we can start to understand how individual cells are interacting with each other or form larger tissue areas across the tissue. So next, I'm going to highlight a couple of key advantages of MERSCO platform. And there are many different spatial genomics platforms on the market now. And I also wanted to very briefly mention how MERSCO differentiates from the other platforms. First, as you can already appreciate from the raw Murphy's data that I displayed, Murphy's is a very quantitative measurement, and it's also very sensitive. So on the left, this is a comparison of Murphy's versus single molecule fish. Each dot is a gene. About 10 genes were measured here in this PNAS paper. Murphy's was able to recapitulate about 94% of the transcripts detected by the single molecule fish, which is oftentimes considered as the gold standard for quantifying RNA copy numbers. So very quantitative and very sensitive. And this sensitivity actually spans several orders of magnitude for different level of gene expression. On the right, this is a measurement in situ, actually in a tissue sample. Each dot is a gene, and there are more than around 500 genes measured in these experiments. And as you can see, we're comparing and benchmarking with bulk RNA sequencing. The x-axis is the FPK value here, and y-axis is the copy number per cell measured by Murphy's. So Murphy's was not only able to measure genes expressed at a very high level, but also with FPK down to 0.01 copy number, and correlation coefficient is 0.88. A very, very nice linear correlation here for not only low, but also high genes, and meaning that this is a quite accurate measurement for a very large range of expression for profile genes. Secondly, Murphy's has exceptional detection efficiency. So in order for us to do imaging, oftentimes we do care about how much signal we're detecting and how many basically counts that we're getting from each measurement. So this is coming from a measurement from two different kinds of spatial genomics approaches, Murphy's and array-based platform. And at the bottom here actually highlights where the data set is coming from. The array-based platform is coming from 10x Genomics. And this is showing one of the marker genes, a median level expression gene, OPRD1, from Murphy's versus array-based platform here. And hopefully you can already see the striking difference in terms of detection efficiency. With Murphy's, you can clearly see where OPRD1 is expressed across the mouse brain. And you can even zoom in and notice, for example, here, a couple of cells expressing tens of copies of OPRD1 in the hippocampal region. However, at the bottom, with the array-based platform, there's only a few grid spots. And each grid spot is around 55 micron, and in between, there's not much information. So thereby, there's no true single-cell resolution. Furthermore, by comparing the counts coming from these two measurements, we found that Murphy's was able to capture 70 times more transcripts per gene detected versus the array-based platform, highlighting the sensitivity here. As mentioned, by leveraging the cell segmentation algorithm and assigning transcripts to individual cells, it enables us to do true single-cell atlasing. So in this science paper, using 155 genes, the authors profiled the mouse hypothalamus region using Murphy's, and it is actually one of the very first demonstrations of Murphy's for cell atlasing. So in total, they profiled around 500,000 cells, and about 75 different cell types were discovered, were found in this brain region. The authors were also performing single-cell sequencing in this study. And back in 2018, there was a lot of uncertainty whether targeted imaging approach like Murphy's can recapitulate all the cell types found by transcriptome-wide technology, such as single-cell RNA-seq or not. And at the bottom left, hopefully you can actually see, Murphy's was actually able to very nicely recapitulate all the cell types found by single-cell RNA-seq, and in certain cases, even resolve cell types that are not discoverable by single-cell RNA-seq, mainly driven by these lowly expressed neuropeptides that can distinguish different cell states. And you can kind of see a couple of Murphy's clusters that are grouped into a single cell cluster here at the bottom left. But more than that, Murphy's provides spatial information. Now, researchers will be able to map out where individual cells are. So now projecting from anterior to posterior of this brain region, you can hopefully see where individual cells are now. And by including cells that are actually, including genes that can report cellular activity, in this particular case, a gene called C4, researchers were able to now figure out which particular cell type are responding to social behaviors such as parenting or mating behaviors. And now they can figure out where exactly these cells are across the different brain region. And now you can start to really imagine the profound implications of this study, because now we can zoom in into a specific location of tissue and pinpoint which cells are responding to certain external stimuli or social behaviors or so, or even drug. Furthermore, Murphy's data can be integrated with single-cell RNA-seq data, and that actually allows transcriptome-wide spatial profiling. So this is a type of sort of analysis pipeline that we continue to see very often now nowadays. Basically, by imputing data from single-cell RNA-seq, researchers will be able to sort of actually measure and predict the expression and spatial location of genes not included in a Murphy's panel, and that will allow oftentimes, for example, a tissue-wide, a transcriptome-wide ligand receptor interaction analysis with spatial context. For example, in the brain region here, what I'm showing here is sort of a ligand and receptor interaction across the brain region. And highlighted here are D2 MSN neurons interacting with itself and cholinergic neurons through a PANK and OPRD1 ligand receptor pair. And with Murphy's data now, because it has spatial context, researchers will be able to directly zoom in into tissue region and actually see where those cells are. As highlighted here, you can zoom into brain region, and you can see that the D2 MSN neurons are expressing both the receptor and ligand, showing in red and green dots, while cholinergic neurons are only expressing OPRD1 transcripts. While on the other hand, D1 MSN neurons are not expressing any of those, as highlighted in these blue marked cells here. So truly highlighting the power of ingesting and imputing data and creating transcriptome-wide single-cell data. Furthermore, the platform has very high cell throughput for those who are running single-cell sequencing. Potentially, if you're to run, say, 1 million single-cell sequencing, the cost of running such a say will be sometimes a few $100,000 or so, very costly. However, for image-based assay, in order to capture more cells, it's fairly straightforward. We just need to tie more field of views to image. And for Murphy's, a square centimeter sized tissue can be captured with hundreds of thousands, sometimes more than 1 million cells can be imaged in a single run. So all of a sudden you have more data and more cells gathered in an experiment. Furthermore, the platform is compatible with protein co-stain. Showing here is human colon cancer, FFP, human colon cancer imaged by 347 genes and 3 proteins, and highlighting sort of the morphology captured by the protein staining here. And on the same tissue slice, you can actually overlay the transcripts captured by RNA imaging, and hopefully you can kind of really see the beautiful overlay image with protein and RNA on the same slice here. The platform is widely applicable for different tissue types, including cultured cells, fresh frozen, fixed frozen tissue blocks, FFP tissue blocks as well. And on this slide, I'm highlighting 6 different tissue types. Actually, all of them are coming from FFPE, measured by a panel of 244 genes. And only a few marker genes are shown here because it's very difficult to show hundreds of color imaging. And within Vizgen, we validated the assay in more than 50 different tissue types, truly highlighting the how versatile this platform is across different disease areas. Furthermore, the platform has incredible flexibility for different sample types, including TMAs. This is a study coming from Medical University of Vienna, where they used a human melanoma tissue array and used an immuno-oncology panel, 500 genes, and 106 cores were measured in a single run, coming from more than 100 patients. You can kind of see now how much data that you can get from a single experiment. And the platform also can annul completely custom panel design. You can select whichever genes you wanted to image for your research of interest. And it's compatible for any species beyond human, mouse. We have now done, for example, rats, monkey, axolotl, xenopus, plants, and many other species. As long as it has well-annotated transcriptome, it's doable. And it can go up to 500 genes currently, and with 1,000 gene custom gene panel coming soon. At the same time, for researchers who may not want to design their own gene panel, we're also offering predefined gene panels that you can actually just directly go, and there are already genes that are coming in, and there will be more gene panels like this coming as well in the future. Furthermore, this is also technology very rigorously validated in the field. The first publication of Murphy's came in Science back in 2015, and now there are more than 150 peer-reviewed publications and preprints available now. And showing here, you can see actually in 2023, the publications are already way bigger than 2022. And then the impact factors showing here in this bar chart are actually very high. More than half of those are actually Nature, Cell, or Science, or the subjournals. And many institutions around the world are already using Murphy's or Merscope. That's a couple of them are highlighted here. So in terms of applications, you can imagine that Murphy's can be widely used in many different research areas, and highlighting, for example, key applications in neuroscience, including mapping out where cells are and figure out what is the function of those cells in aging, in memory, or in disease progression. Yeah, and later I'm going to highlight a couple of studies in detail. Then similarly, in oncology, we have seen, for example, across industry as well as academic institutions, for example, to discover biomarkers in tumor, to figure out how oncology checkpoint inhibitors, for example, PD-1 treatment, and what is the mechanism of action for PD-1 treatment in liver cancer or in lung cancer, and then also to figure out some novel targets in ovarian cancer and colon cancer on the right here as well. A third big application that we're seeing, for example, is in infectious disease, for example, in liver infection, in brain infection, and pathogen attack in plants as well. And beyond this, we actually have seen a lot more applications in kidney research, in breast cancer, in breast and normal, even in hearing and in many other areas. I'm just going to highlight a couple of more studies in the last few minutes here, just to really speak for what type of research questions can be addressed by this technology. First, in neuroscience, so as we all know, brain is a tissue that has many different cell types, and how many cell types there are in the brain, where they are, what the cell states are, how do they interact in the brain to form high-level structures are not so well known, and how does the brain function, what is the mechanism for some of those diseases also not so well understood. So to address these questions, we need a tool like Murphy's that basically can map out these building blocks in the brain and then characterize the function. So in this area, we have seen a lot of really cool publications for Atlas in basically building these reference maps, starting from the very first one, the Science published in 2018 to now a very long list of publications across different research groups. To functional studies, actually to figure out how these cells function or so. I'm going to highlight 2 to 3 key studies in this area. First, on cell isolation. This is a study mainly done by Harvard University and Allen Institute. And the key goal here is to create a reference map of the mouse brain, basically to figure out where those cells are, how many cell types there are. And in this study, they performed single cell sequencing for about 7 million cells and did Murphy's for around 4.3 million cells from Allen and 8 million cells from Harvard. And they constructed the first high-resolution spatial cell atlas in the whole mouse brain. Basically, every 100 microns, they cut the mouse brain slice and imaged by Murphy's. And then through this very comprehensive study, they characterized the roughly around 5,200 different cell types and states in the adult mouse brain. Here, you're seeing a very, very complex UMAP highlighting all the different major cell classes or subclasses and supertypes here. And with that, the researchers were also able to integrate single-cell RNA-seq data with Murphy's data, enabling transcriptome-wide spatial profiling. This allows them to systematically characterize the distribution of all neuronal and non-neuronal cell types, neuronal transmitters, neuropeptides expression. Highlighted on the right, you can kind of see where individual cells are located now. And that also allows them to investigate cell type-specific interactions between molecularly defined cell types. So this will truly be a foundational paper and really great resource paper for anyone in the neuroscience community that are looking for this type of data. And this is now publicly available, these data sets from Allen Institute. So the second short study, this is coming from Thomas Sudolph's lab and Stephen Quack's lab at CCBio Hub in Stanford. And the key question here is to figure out how memory is formed. And they used a mouse model that are sort of for fear recalling, and they studied a particular brain region, amygdala here. And in this particular case, they have a genetically modified mouse. When the mouse recalls the fear, then it basically expresses more TD2 matter. And as you can see from this picture here, when the animal are recalling the fear, indeed, there are more cells expressing TD2 matter here. However, where those cells are, what is the identity of the cells is not very well understood. Thereby, they used Murphy's and constructed a few hundred gene panels, mapped out all the cell types in this particular brain region. Shown on the top left, you kind of see all the molecularly defined cell types here now. And then by doing fine subclustering of those cell types in this particular region, the researchers were actually able to find a unique subtype of the cells that are PANK positive and TAC lective, which is highlighted here. You can kind of see the differential gene expression in this particular cell type, a particular cell type that are PANK positive and TAC lective, that seems to be essential for long-term memory trace. And then by examining and performing neighborhood analysis, what the researchers found is that the interaction of astrocytes in the brain with this particular PAINT1 and TEC1 tech-elective cells are very important for long-term memory consolidation and disruption of this actually leads to a loss of that. And truly highlighted now how you could potentially use this to understand cell-cell interaction, and that cell-cell interaction is essential for memory formation and consolidation, highlighting the power of spatial data for understanding how brain cells are functioning in the brain. The last example in this here, I'm going to just very briefly show that basically how researchers at any institute are using this to build a multimodal cell aspects of AD. Alzheimer's disease is a very complex disease, and over the past few decades, the efforts to actually develop a drug has largely been very, very disappointing until very recently. There has been at least therapeutics now being on the market. And in order to understand how AD progresses, it is important to understand the building blocks of the brain, how it is changing at different disease stages. So Alan took an effort to actually using the human patient data, actually did a multimodal studies. They used pathological scans to scan all the hallmarks such as A beta, synuclein, and so to figure and use this to annotate the stage of the disease. Then they performed single nucleus ATAC-seq, multi-arm single nucleus RNA-seq to get single cell data. For spatial transcriptomics, they chose Murphy's assay here to get the spatial data. Thereby, it allows the researchers to integrate everything together. And at the bottom left, hopefully you can kind of see the high correspondence between MERSCOPE data and single nucleus RNA-seq data with all the cell types found. It enables researchers now also to integrate single cell and single nucleus data with Murphy's data, thereby predicting the location of the genes that are not included in Murphy's assay. And with all that, then it helps the researchers to figure out all the different cell types in the human brain, in this particular part of the human brain, and then at the same time figure out how the cell types are changing as the disease progresses. So that allows the researchers to answer questions that couldn't be answered for many, many years. For example, this spatial temporal analysis of cell type proportion during AD progression. Highlighted here, from left to right, you kind of see the progression of disease from early to late stage. And very interestingly, as we examine this, a lot of neurons, as expected, at later time points, because AD, the brain will shrink, then there will be loss of neurons. Indeed, we see a lot of neuronal types such as excitatory neurons or probably positive lectin inhibitory neurons started to decline at much later stage of the disease. However, a subtype of the inhibitory neurons, that is SST positive, seem to have a much sharper decline at very early onset of the early onset of the disease, as you can see by this orange line here. And this now can be very easily visualized in space. For example, highlighted here at the bottom right, you can kind of see excitatory neurons. Until much later stage, you started to see the decline at different layers of the brain. However, SST positive inhibitory neurons, you see a very sharp decline from early to middle stage of the disease, while in contrary, probably positive inhibitory neurons doesn't behave in that way. So basically, they found this early reduction indicating that maybe SST inhibitory neurons might be quite important for the and susceptible for the AD disease. So hopefully these few studies sort of actually captures how researchers were using this to build reference map, figure out how cells function and what is the mechanism of some of those neurological diseases. And I'm going to sort of move on to two examples of oncology and how MERSCOPE has been used in understanding how the mechanism of action for drugs. And here, many important questions, because cancer, again, is a very heterogeneous disease, and how different biomarkers are displayed during different stages of the disease, how different cell types are interacting with each other. We just heard from the previous talk, for example, different cells are attacking each other. How are they interacting in situ? Now with a spatial assay like this, now we can really examine this type of spatial interactions. So in the first study, this is now published in Nature Medicine online now, researchers at Mount Sinai used MERSCOPE to sort of understand and study the mechanism of action for PD-1 blockade in human liver cancer. And in this particular study, it's a phase clinical trial study. They collected samples from pre-treatment to later treatment. And as in this clinical trial, about 15 to 20%, 25% of the patients are responding to the treatment, while majority are not responding. So the researchers wanted to understand why so only small fraction of the patients respond. And then they examined and stand the CD3, and you can see there's two major phenotypes: T cell reached, infiltrating in the tumor, T cell excluded, many residing in stromal region, and the T cell low phenotype as well. The responders, many exhibited the T cell reach, while the other two phenotypes are more in the non-responders. And as the researchers performed single cell sequencing and some of the other assays, they found that actually there seems to be expansion of CXCL13 positive CD40 helper cells and PD1 high effector CD8 T cells in responders. Hopefully you can kind of see here, actually in the responders in this blue bar here, there are not more. And when there's more CD4 T helper cells that are positive for CXCL13, oftentimes you see more of the CD8 PD1 high effector cells. And since CD4 T helper cells can really help prime the CD8 progenitor T cells, and it makes the researchers wonder actually are CD4 T helper cells and the progenitor CD8 T cells actually interacting with each other locally, and that triggers the expansion of CD8 T cells during the progression of the disease or after treatment or not. And in order to answer such questions, a spatial assay is needed, and that's why the researchers turned to use Murphy's here. They designed a 500 gene panel and then actually used this to figure out all the cell types in the human liver cancer sample, and then through proximity analysis, they were able to sort of figure out how cells are residing in the tumor. And one very interesting part, I will just highlight this picture here. Basically, they were able to find this more like immune hub type-ish clusters of immune cells where B cells and dendritic cells are residing inside and then surrounded by these progenitor CD8 T cells and T helper cells here, and activated dendritic cells are even outside. And of course, with many other supporting data coming from this study, it helps the researchers to come up with a model where the dendritic cells are presenting to T helper cells here, and that helps to prime the progenitor CD8 cells and in responders, and that makes them to proliferate into a killer effector cell to kill the tumors, while the non-responders don't seem to have this local enrichment of immune cells, and thereby it doesn't actually seem to be able to kill the tumor cells effectively. And lastly, the example here tells a somewhat similar story, but slightly different angle. This is a study coming from Mass General Hospital from Neil Cohen's group, where they were interested in studying the mechanism of action for PD1 in human lung cancer. And this group back in 2017 published a Cell paper identifying a so-called immune hubs that are actually very much positive for the chemokines CXCL10 and 11, the macrophage secreting this type of chemokines here, as you can see from the staining here. But what type of cells they are in these immune hubs is not clear, and what cells are driven the secretion of these chemokines and what are the cell-cell interactions is not super clear. And very interestingly, having this immune hub actually is associated with much favorable outcome in PD1 treatment. You can see the responders usually have a much larger, more immunity hubs. So again, to understand the cell types in this region, the gene expression profile within the hub versus the non-hub regions, the researchers turned to use Murphy's, and then they designed the 500 gene, actually 479 gene panel, and then performed two type of analysis. First is that they did molecular atlas in here, figured out the tissue region that are positive for this CXCL10 and 11, segment those tissue regions, and then perform differentiation gene expression to figure out what type of genes are expressed, what group of genes are specifically enriched in immunity hubs versus non-immunity hubs. Separately, leveraging the cell segmentation possibility with MERSCOPE, then they were able to perform downstream single cell analysis and actually figure out all the major cell types and specific substates of the immune cells, and then do cell-cell interaction analysis. So at the bottom is a few key findings. For example, profile gene expression in immunity hubs. Hopefully you can kind of see here now in immunity hubs, the first column, there are a lot of key chemokines that are enriched now as highlighted versus the non-immunity regions, such as CCL19, CCL17, 22, and others as well. At the same time, they were able to perform single cell analysis and figure out the cell type composition here. And very interestingly, stem-like immunity cells that are positive for TCF 7 are enriched in the immunity hubs, but depleted in the non-eastern immunity hub regions. And with all the different state of the immune cells figured and actually mapped out by Murphy, now researchers were able to perform neighborhood analysis to figure out how cells are interacting with each other within the eastern immunity hub. And this heat map sort of shows the interactions within the immunity hub. You can kind of see actually dendritic cells and CD4 T cells, Tregs, B cells, and these stem-like CD8 T cells, positive for TCF7, are actually quite enriched. Again, enabling the researchers to put together this very nice model where dendritic cells are presenting and recruiting to CD4 T cells, and then it starts to secrete the chemokines, and that recruits this TCF7 positive CD8 T cells, and then that further triggers the proliferation of the cells and then makes them to become effector cells to kill the tumor here. Again, a very nice model and highlights the power of Murphy to actually figure out the mechanism of action for PD-1 treatment in lung cancer here. So I will just conclude here with the last slide, just summarize the key advantages of Murphy, and hopefully the studies highlighted in the presentation now convince you that Murphy is a very powerful platform that enables multiplexed imaging of hundreds to thousands of genes in a single sample. So it can also enable protein co-detection, and you can use custom gene panels and design gene panels for your research of interest. It has very high resolution from whole tissue to single cell down to subcellular resolution, has extremely high sensitivity and specificity for detecting RNAs, has very high throughput, knowing hundreds of thousands, sometimes more than a million cells without the need of sequencing. And then it also has extreme flexibility to run on many samples or tissue types. So with that, I will pause here and let me know if you have any questions.
Thank you very much, Jiang. A great overview of the technology. We have not a lot of time, a few minutes for questions. We have one from, actually, before we go into questions, just wanted to announce we'll have a networking social, and we're very grateful to Vizgen for supporting that. It's at 5:30 p.m. today at Bodacious in Biopolis. All are welcome to join. And you can ask the speaker, who will hopefully join as well, more questions over there. All right. First question from Tim. When you have over 5,000 genes from, over 5,000 cell clusters from 500 genes, then how big are the differences between the clusters? It should be very subtle, right? And can you really validate such subtle differences between cell types?
Yeah, I think this is a really great question here in terms of the sort of cell type and what is actually defined as the cell. And actually, in this field, this is always an ongoing discussion. But I think in terms of the difference, there are quite a few things that, firstly, transcriptome-wise, there will be differences. But at the same time, the location of the cells can also really help tell some of those differences. So it really depends on how the cells and states are classified. And I think it's a lot of nuances over there in terms of that level of clustering. So
yeah, I'm going to push you a little bit on this because, and here I don't mind inserting my personal opinions. When we start talking about thousands of cell types, it's just not validatable. At most, in one study, you can validate one cell type. But even there, it's hard, like Tim was saying, because the neighboring cell type is so similar to the cell type you're validating when you have 5,000 cell types. I wonder if it's even meaningful to call them cell types or even cell states. And if you change the clustering algorithm, you'll get a different 5,000 cell types or cell states that will very loosely correspond to these 5,000, but it'll be a very, very noisy correspondence, right? So is it really meaningful to claim that there's any biological identity for 5,000 distinct units if they're so variable and unstable across clustering algorithms and spatial omics assays?
Yeah, I think this is actually also potentially tied to the particular research area as well. I think in oncology, we probably don't usually hear or sort of actually see so many cell types actually identified in a tumor sample or so. But in neuroscience, for example, a very small region like hypothalamus or so, researchers can, like back in 2018 and across many different regions, easily identify hundreds of cell types over them. So it's not too surprising for a structure like brain that you can actually very finely define the cell types over the states, because the brain is very, very finely organized in such a way and depends on the location, transcriptome states. So I think it's kind of depends on the research area as well as how you classify the cells in different disease areas or research areas.
That's precisely the problem, isn't it? You can classify the cells any way you want and get any number of clusters. 5,000 is not an upper limit. With the same data set, they could have increased the resolution of the clustering algorithm and gotten 50,000 clusters. There's nothing unique about 5,000. I'm sorry to push back so strongly against this, but I feel it's harmful for the field for people to claim that there is something unique or special about their 5,000 clusters or somebody else's 400 clusters. These are just carving out a continuum into regions, you know. It's like saying Europe has 72 countries or whatever, however many countries Europe, like 30 countries. That's arbitrary. Some human beings decided to draw a boundary here. Europe could have had 500 countries if you drew boundaries more finely and created smaller countries, right? And similarly, you can have that any number of excitatory neuron subtypes. You just have to you have to create smaller countries, and then that continent will have more countries, you know?
Mm-hmm. Yeah, I think downstream, it's a really, really good point here. And I think it's exactly where this field is evolving. And because those cell types classified, they actually, whether it's carry unique functions or the display in very unique spatial locations and all that, I think the first level is actually to understand sort of these unique types and states, and then map out those building blocks first as a step. And I think in a way, it's kind of an evolving field here as well, for sure.
Fair enough. And I just want to make it very clear also, I'm not holding you personally responsible for what those researchers wrote. So, I mean, they certainly benefited tremendously from the Morphish technology. And I think that data set will be a foundational resource for a lot of people, just as the Morphit 2018, I think, mouse brain data set was a great resource for the community, and we have used it as well. Question from Jagdish, very nice presentation, thank you. Curious to know your thoughts about the usefulness of super resolution microscopy for spatial omics.
Yes, so as of now, Morphish is not built based on super resolution microscope. It's actually a regular epi already, because we're doing single molecule imaging, it already contains some level of super resolution information inside due to our decoding algorithm.
Absolutely. So some people have extended the definition of super resolution, but yeah, you're absolutely right. Tim was chiming in again on the concept of number of cell types, but I'm going to switch to another topic if there are no other questions on the chat. Quite a few topics actually. So I'll come up with the one thing that always comes up when people who are not from companies talk about spatial omics, and I'll surface it to you, which is cost. So as you know, there's a lot of excitement, and I'm sure you're selling these machines faster you can make them. But all your users will be complaining to you about cost. And really, like, what do you see about the likelihood of that becoming more affordable for large-scale studies?
Yeah, so I'm
and this is not Vizgen specifically, it's the whole field. Okay, your competitors are also too expensive.
On the price here in terms of this, but in terms of the average runs, it's roughly very similar to the single cell assay, and as we are seeing how the single cell field evolves, right? So over time, the run per cell is continuing to decrease, and I imagine that will be a very similar pace for spatial genomics field as well, because this is also a new emerging field, and technologies are actually getting improved and upgraded every once in a while. So.
Thank you. That's exciting. And again, I want to emphasize cost is high for all technologies. So yeah, I'm excited to hear that it'll be coming down just like the single cell omics cost per cell came down. And then gene panel, it was great to see you mentioned 1100 genes for the mouse brain panel from Harvard. But I guess that's like more custom panel of theirs, right? Or would that be commercially available?
So we will have up to 1,000 custom gene panels commercially available very soon. So it's like you can select whichever gene you want.
Fantastic. And how much 3D info do we have in this data?
So it does have 3D information. We usually image about 10 macrons and in 7 Z slides. So it contains the Z information.
All right. And so do you provide 3D cell segmentation or is it 2D?
So the cell segmentation wise it's kind of captured mainly it depends on the algorithm that is applied in the data sets. Yes.
But the default Vizgen cell segmentation algorithm, does it give you a 3D segmentation or 2D?
Mm-hmm. So for example, our watershed algorithm, it contains the 3D information, and you can actually see in our publicly released data sets, the cell segmentation is every Z slice, it has the cell segmentation on the shape of the cell. But of course, we also nowadays released sort of a custom version of the cell segmentation that users can sort of plug in the on parameter and do so. So in those ones, so basically the default, for example, using watershed is 3D, but if you choose the one by yourself, not using the default, you could also just do the middle plane cell segmentation to speed up the analysis. So it's kind of yes to both.
Okay, that's great to hear that there's some flexibility. So, and you mentioned that incredible mouse brain data set, mouse brain morphish data set. Is there a comparable human brain data set that we can access soon?
I believe Allen Institute is going to also make the AD data publicly available. So I think coming soon.
Okay, looking forward to that. One last question. You showed a beautiful benchmarking figure showing linearity over 4 orders of magnitude, maybe even slightly more than 4 orders of magnitude between the morphish readout and I think FPKM and bulk RNA-seq. It's quite impressive to be able to detect expression over such a big range. I'm curious to know what was the sample type and how big the data set was.
Yeah, so that was a mouse brain data set. It's actually publicly available from Vizgen's website. If you're interested in checking the data sets, it's downloadable from there. It contains 483 genes. In total, the data sets has 9 different brain regions, 3 sliced from 3 animals, so 3 by 3. But what I was showing is just coming from a single data set out of the 9 measurements.
Okay, fantastic. I think I know which data set you're referring to. Yeah. Okay, great. We're slightly over time, actually. Very grateful to both our speakers, Lih Feng and Jiang, for a very stimulating session. Hope to see as many of you as possible at the networking session today at 5:30 p.m. And we have a message from Anjana Narayanan in the chat saying Singapore attendees can come to Bodacious in the evening, learn about current products, upcoming products in person. All right. Thank you once again. And hope to see you all again soon.
Thank you. Bye.
Thank you.